All right. Yeah. So my name is Andrew Leduc, and I'm a third-year graduate student here at Northeastern in the Slavov lab. And so welcome to the first of three more focused talks on different aspects of the single-cell proteomic sample prep pipeline. Am I doing this right? Sorry, everyone. Yeah. This clicker work for you?
Yeah.
Oh, there we go. Okay. Sorry about that. So in this talk, we're going to be primarily talking about the sample prep side of the workflow, and in subsequent talks, you're going to hear a little bit more about some of the more downstream parts of the process. And so just to give an overview of some of the things that we're going to be talking about here, first, we're going to address important different features that we'd ideally like to have in our sample prep, and then talk about different approaches that the field has taken. There's been kind of an explosion in methods for preparing samples by single cell proteomics by bottom-up mass spectrometry. And then we're going to talk about different ways of evaluating the quality of the data that you get from these sample preps, which can be a bit tricky. So it's good to kind of outline important ways that this can be done, kind of regardless of the method that you use to prepare your samples. And so those of you who don't know, here's kind of a very high level picture of all the different steps involved. And so we obviously start with our single cells. We need to isolate these cells somehow so that we can perform reactions on each cell individually. We then need to lyse our cells and denature the proteins, making them available for enzymatic digestion, where they can then be labeled or left for multiplexed analysis or analyzed label-free. And we just lastly need to collect these samples so we can deliver them to the mass spec for downstream analysis. And so, as you can assume, there's a lot of different choices that you have and how you go about performing the different steps in this workflow. But regardless of what choices you make, there's a couple things that ideally we'd like to have as kind of guiding principles, nice to have features of our sample prep. And I've just kind of outlined three here, but of course there's more. The first being accessibility. We really ideally would like our methods to be accessible so that we can get single cell proteomics into the hands of as many people as possible and empower more discoveries with this technology that we think is really cool and potentially powerful. And the second is throughput. So we want methods that allow us to prepare a large number of single cells so that we can fully take advantage of the promise of single cell analysis, which is really to explore complex samples, differentiate all the cell types that are involved, and also sample sufficiently continuous differentiation processes. But we don't just want to classify these cell types, like Nikolai said earlier. We want to be able to make quantitative measurements so that we can examine the distributions of these proteins and these underlying processes, and we can start to learn a little bit more and infer potential regulatory interactions and start to learn more about these networks of proteins. And so to address these different desirable features we'd like to have, the field has made substantial progress recently, and these steps have been empowered by a couple of kind of themes, one of which being using approaches that are use reagents compatible with LC-MS analysis. Like Nikolai said earlier, the instruments can be a bit sensitive and finicky to contaminant reagents. So using things that are compatible with mass spec obviates the cleanup of thousands of potential samples that we'd like to analyze and can help increase the throughput of the sample prep. And similarly, automation has been used along with minimizing volumes to improve throughput, remove user error from the sample prep process, and reduce the amount of contaminant reagents that we might have in our samples. And last, the creative designs from the field that have allowed for this throughput to be scaled up through various different means. And so for the purpose of this talk now, to get into kind of specific approaches taken, we've categorized the sample prep approaches out there into kind of four categories. Although this is how we've categorized them for the purpose of this talk, you can probably categorize them many other ways. But to start, we're going to start with kind of the simplest, which is taking single cells and preparing them in individual tubes. And sorry. Okay. And some early work along these lines came from Peter Nemes's group looking at xenopus oocytes, where samples single cells are sampled by small capillaries and then subsequently prepared. Also work from John Yates, which you heard about maybe a previous year's SCP, using patch clamp to extract the component proteins from neurons, in addition to making electrophysiological measurements and then preparing these cells individually. And also, there's early work from our lab, Scope MS, which for us was really nice as a kind of proof of principle for single cell proteomic sample preparation. And here in the early days, cells were either handpicked or sorted by fax machine into individual tubes into volumes of about 10 microliters. And then these tubes were put into these Covaris tubes that could sonicate them to provide a lysis method that was not adding any reagents. Again, that would be poisonous to the mass spec. And like I said, this work provided a nice proof of principle for single cell proteomics for us, allowing us to kind of finally analyze small cells. Here we're showing some monocytes and T cell lines, and also have measurements that were validated externally by different metrics such as fluorescence. But again, of course, taking every single cell individually and lysing them one at a time is not very scalable if we're interested in preparing thousands of cells. And so this kind of brought in the next wave of approaches that instead of preparing cells in individual tubes, we can take those tubes and kind of compress them to the many wells of a multi-well plate. And so towards this end, the Scope 2/mPOP methodology was developed by Harrison Specht in our lab, and this allowed for kind of a scale-up in sample preparation by sorting into the many wells of a 384 well plate, and then providing a lysis method that scaled past just doing one cell at a time by using freeze heat to lyse all the cells in the 384 and 96 well plate simultaneously. And as well, it introduced further automation with systems such as the Mantis that allowed for quick dispensing of reagents, and also to take work out of the hands of the users interested in preparing these samples. And so this allowed for the first single cell proteomic study with over 1,000 cells, where they not only were able to differentiate two cell types, which you could just look under a microscope, of course, and see the differences quite easily, but they were also able to detect gradients of heterogeneity within one population, macrophages, in the absence of polarizing cytokines, thus taking kind of advantage of some of the promises of single cell proteomics, which is to detect these gradients of changes in protein abundance. And if you're interested in learning more about nPOP and this method, you can go to our website. We have tons of resources. There's a protocol out there, different video tutorials and such. And another method along these lines from the Kelly lab was Autopots, which also prepares samples in 384 well plates and uses further automation systems like the Opentrons that can allow, just past the dispensing of reagents, can also allow you to combine samples, and has this nice additional layer of automation that can further speed up the rate of samples being prepared. And so getting back to these points that we talked about allowing for advances in the field of single cell proteomic sample prep, another one that was of intense interest is minimizing volumes, again, for reducing the amount of potential contaminant reagents, avoiding losses of proteins to being exposed to larger surface areas. And so some really unique approaches to solving this problem were to miniaturize these wells past what you have with the 384 plate and with the use of microfabricated chips. And some early work providing data along these lines was, again, from the Kelly lab, the nanopots methodology, and also oil air droplets that took these reaction volumes down from past like the 1 microliter range into the range of nanoliters. And there's been additional recent work along these lines, scaling up the sample prep further and reducing volumes with additional chips down to volumes of about 20 nanoliters, and allowing for a scale up in the number of single cells that can be prepared simultaneously with the N2 method developed by Ying Zhu, and also with the ProteoChip. And so these samples also rely on the Cell-in-1, which is a really gentle cell dispensing and liquid handling system that allows for these processes to be automated further, and allows for this fine precision that can allow you to reduce volumes and spatially take advantage of kind of fine grained XY coordinate dispensing past what you'd have with a fax sorting instrument. And so one of the additional advantages of minimizing sample volumes is that you're also able to and scale throughput by increasing the density of samples over the area of your sample prep. And so our approach to doing this was to prepare samples on the surface of a glass slide that has a fluorocarbon coating, but has kind of no constrictions on where you can dispense samples over the surface of the slide. And so to these ends, we developed the nPOP method, which again takes advantage of increasing throughput and advantage of these small volumes, as you can see in the lower right-hand corner, by kind of being able to further squeeze these samples into kind of tightly into these small arrays. And so one of the also additional benefits of having this freedom is that we're able to have a sample preparation that is flexible to different designs. So whether you're interested in doing, 16-plex with TMT or 11-plex, or different labeling schemes such as the multiplexed DIA workflow, which has 3 labels, or even you're interested in doing label-free analysis, having this flexibility can allow you to prepare a large number of single cells regardless of this design, and allow you to prepare over 2,000 individual single cells in a single day of sample preparation. And so I guess don't just take my word for it. Along these lines, we've published data on utilizing different multiplexing schemes on different cell culture samples, as you saw some of the earlier data from this Plex DIA method with the 3 different cancer cell lines, and also in our nPOP preprint, where we prepared over 2,000 single cells in a day, and which kind of made it to the end of the analysis here was about 1,500 single cells that you're seeing in the plot on the right-hand side. But in addition to preparing samples just from these cell culture samples, we've kind of moved past this into primary tissue, primary cell samples. Some of this work early on was from Huffman's prioritized scope approach, where we were analyzing single cells from primary bone marrow-derived macrophages. We've also analyzed samples from murine-dissociated lung tissues. And further, we've recently analyzed over 5,000 single cells, as Nikolai alluded to earlier, in a project led by Saad Khan, where this was a nice success for the nPOP sample preparation method, because previously we had a great deal of difficulties preparing these samples isolated by the fax machines, and there was kind of a high level of contamination, and it was difficult to disseminate signal from the protein that we were measuring from contamination. And so these are all advancements that have been enabled by these small volumes in this flexible design. And if you're interested in learning more about nPOP, we've tried to make some additional resources available on our website, tutorials and videos. In part, there's been interest from the field in pursuing these different designs, so we've tried to address these with different resources. And hopefully later in the conference, you might hear some data from this method being used outside this lab from David Perlman. So those are kind of an overview of the different approaches that are out there for preparing samples by single cell proteomics by bottom-up MS. And so after choosing a sample prep that you've decided best meets your needs, the next question is, did my sample prep work? How is this data going to provide the quality that I need to make the biological conclusions that I'm interested in? And so kind of the first big point along these lines is what in RNA-seq they commonly refer to as the capture efficiency, but really we're talking about how much of the proteins in our single cells are we ultimately able to deliver to the mass spec for analysis. And so this question can be a bit hard to totally understand, since different mass spec instruments and different methods and chromatography setups can give kind of a wide array of different results. But one of the easiest ways to do this that we've kind of taken advantage of in our lab for kind of benchmarking this is to prepare samples in bulk in methods that you have a little bit higher degree of confidence don't result in large losses. And what you can do is you can take these bulk samples that you prepared and have confidence don't arise in a large degree of sample prep losses, and you can dilute them and compare these abundances to the abundances of the single cells that you've prepared. And so this is exactly what we've done here for our published nPOP data set, where we're comparing the intensity of our diluted single cells to the intensity of our single cells to the intensity of diluted samples down to the 1 cell level. And we see that this distribution is centered around 0 on the log scale, and it's about around 96% of the intensity of diluted samples. And so past that, additional ways of kind of evaluating how much protein that you're extracting from these cells is you can utilize orthogonal metrics. And here we're taking advantage of the fact that the cell in one, which dispenses the cells, also records the diameters of the single cells. And so what we can do is, for the most part, we expect if the concentration of protein is scaling with the size of the protein, we expect the total protein content to scale with this diameter of these cells. So what we have in this plot on the far right is that we've plotted this measured diameter against the measured protein content and for each individual single cell. And in general, we see good agreement, though in all cases, you might not expect the concentration necessarily to scale with the cell type, and you'll hear a little bit more potentially about that later in the conference. Another important metric, like I've been kind of talking about for the majority of this talk, is keeping the contamination low. In mass spectrometry, ions that have a charge of 1 are generally coming from contaminants where peptides tend to have charges of +2 and higher. And so we can try to get a sense for the amount of contaminants in our sample versus the amount of peptides we're delivering by comparing the intensity of species with higher than +1 charge to species with +1 charge. And there are other ways, such as the FAMES, to kind of minimize these contaminants in our sample prep, but we're not using the FAMES here. And it's gratifying to see that our samples of our peptides is not kind of being diluted out by a large degree of contaminant signal. So if you have confidence that you're kind of getting strong signal from your measurements, at least in terms of abundance, and that the signal is mostly not contaminant, there's a further question of, is this signal coming from background, or is this really coming from the peptides that we're interested in measuring? And one way that we've decided to kind of go about assessing this in our lab is to include negative controls. So in the sense of an nPOP sample preparation, these are droplets that don't receive a single cell, but receive all of the same reagents. And we're also able to measure the intensity coming from the labels for these droplets. And what we mostly see is that the signal is quite low for these negative controls, which really shouldn't have any signal at all, albeit with some background signal that we see there. And for the signal for melanoma and monocyte cells, they correspond to more expected trends in protein abundance. And so the last point I want to talk about here is the quantitative accuracy of the measurements. And so after you've kind of prepared these single cells, a common way to visualize your data is by dimensionality reduction plots. And here you're seeing a PCA where we're clearly able to kind of separate these two samples that we've prepared. But this is not necessarily because we're measuring differences in the underlying protein abundances. This could be for a number of other reasons, such as technical artifacts. It could be because the missing data is high. It could be because of poor normalization that led to effects due to the cell size. As you saw from some of the earlier plots, the melanoma cells are much larger than the monocytes. And so an additional thing we can do is prepare bulk samples of equivalent protein amount from that might not share many of the same artifacts that could be present in our lowly abundant single cell analysis. And we can take these large samples and we can project them onto our low dimensionality reduction plots. And so this can give us an added layer of confidence that the separation that we're seeing on our PCA is corresponding to changes in the underlying protein distributions to the extent that they might not suffer from all the same systematic artifacts. And just some conclusions and looking forward to kind of summarize the talk. Some things to point out for looking forward is that it's important to benchmark quantitative accuracy for these measurements. And I think there can be additional studies that look at using methods such as spike-ins and different orthogonal measurements to kind of further iron out the kind of extent of systematic biases and random noise that are in our data. And also kind of the last point that we've been making is that the sample prep should ideally be able to scale with the mass spectrometry measurements. We envision a future not so far off where we're able to analyze many more cells with mass spectrometry than we currently can. And, at this current point in time, the sample preparation is not the bottleneck for us in our lab, as it takes about several days to prepare all the samples from one of our sample preps. But we want methods that are able to easily scale as this landscape changes and as the landscape of labeling reagents changes, which kind of we've all seen in past years. And lastly, it'd be nice to kind of explore more studies linking kind of some phenotypic measurements to protein abundances, looking at things such as imaging data, which you have nice photos provided by the Cell M1, but also measurements such as fluorescence that you can receive from FACS, which can be used for different studies, such as in work from Huffman, where they looked at endocytosis readouts. And so there's kind of a lot of exciting things. And these are some challenges that are applied really to being able to do flexible and different sample prep designs. So with that, I just want to say thank you to all the people who made this possible, especially Professor Slavov and all the co-authors on nPOP and everyone in the Slavov lab, and of course to the funders and everyone. So, yep. Thank you.
Questions in the room for Andrew?
Very nice talk, Andrew. It's great to see how far the community's come in miniaturization of the workflow. And I was just wondering what your perspectives are on the limits of that miniaturization. At what point can the LCMS methods still continue to get great data? Could it be in the single digit nanoliter range, or could it be somewhere higher than that?
Yeah, so that's a good question. I think something important to consider here is asking yourself, what the benefits are to the miniaturization, what you're kind of trying to achieve. So, I mean, theoretically, potentially, there's a possibility to scale the sample prep down to the scale of individual nanoliters. But I think the question really is this providing you, does this provide you higher throughput with your data sets? Does this provide more accuracy? And if you look to single cell RNA sequencing, actually some of the methods with the highest degree of capture efficiency are some of the well-based methods, the SmartSeq, that work in volumes of about 5 microliters, and where the droplet methods are actually much more commonly and frequently used. And this is because they've utilized these small volumes to kind of vastly scale up the rate of sample production, and people have kind of been able to do different large scale studies along these lines. So, potentially there is, but I think it has to be kept in mind with kind of the method for just preparing your samples and looking at the various benefits that this could provide, and whether, it's a trade-off of whether it's worth the effort to kind of push those volumes past the already small ones being utilized by lots of workflows.
Alexei Zvyshkin, University of Michigan. Very nice talk. I want to ask you about assessing the accuracy of protein abundance measurements by comparing with diluted samples to say 5 cells. I mean, in some way, we want to study single cells because we say that single cells are different than large populations, right? Mm-hmm. And here you're kind of referring to diluted cells and say, oh, it's good that we have a very low variance that's centered around 1, right? So it's a little bit a questionable way, I would say, right? So I'm not saying it's a bad way. It's probably the best you can do, but if you can comment on that.
Yeah, so I also might have not explained the slide as well as I could have. So I guess there's going to be a high degree of variance for individual single cells kind of corresponding to a diluted bulk sample, as there's a large range of sizes that correspond to the individual cells. So the way we did the measurement was actually to just average out the abundance of kind of all the single cells we measured. So, and this can kind of have a kind of average out some of these differences that you see on the single cell level. Also, yeah, the distribution was kind of a bit broad, which some of that corresponds to slightly different methods being used to prepare the carrier samples, which is kind of with that heat lysis versus the droplet method, which can correspond to, certain proteins being better extracted in one versus the other, which maybe corresponds to some of the tails of that distribution. But in general, in terms of measuring the overall abundance of capture efficiency, I mean, you're not necessarily guaranteed that the bulk methods you're using are totally the best. And I mean, extracting as much protein as possible, I suppose there's always room for improvement. But to the extent that these methods have been kind of benchmarked against other more common lysis methods such as urea or kind of other chaotropic agents, it's a there's trade-offs with every approach to kind of measuring overall protein abundance, but this seems to be an accessible one that we find as a good approach for kind of getting a rough idea, at least in some sense.
Great talk. Quick question about the comparison you did between the two different cell types. So I'm curious, was there the prediction that the distribution that you see in each of the group will be the same or different? And also it looked like that your cancer cell line had a sort of cloud of cells that were different. Is that a real thing?
Yeah. So that's a good observation. We looked at this extensively in our preprint on nPOP, and we think that this cluster represents a subpopulation we kind of tried to validate this by looking at RNA sequencing measurements that have observed similar subpopulations in this exact cell line. So there's always the potential that it could be an artifact of sample preparation, and it's always upon the user and the people doing the work to kind of, to the best of their ability, suss out to what degree these different potential sources of batch effect, be it different days mass specs are, different days your samples are run, different instruments. That was actually a kind of a considerable source of variability in our data. But yeah, that you certainly don't expect those clusters to be totally the same, in the sense that we do think that there's some additional heterogeneity in those melanoma cells potentially coming from a subpopulation. Thanks.
Thanks for the talk. I think this is kind of like a follow-up to your question. So I guess a more general question is that how do you do batch correction normalization? What assumption you're making in these kind of analysis?
That's a great question. So one of the benefits of increasing the throughput and providing additional parallelization to your sample preparation is that you have the potential for less batches that come from preparing cells on different days. And so, in terms of how to implement these batch corrections on your data, I think, you can start by things that you might know to provide potential sources of batch effect, and look at the different variants due to some of these sources, such as samples prepared on different days, run on different instruments. In terms of like the physical implementation of the batch corrections, there's kind of a lot of different algorithms out there that try to minimize variance due to the specific batches that you formulate. So I think a good place to start is looking at just different variances due to different sources of batches that you kind of maybe expect to see in your data. And then there's, some that you can have potential batch effects that are kind of hard to explain. And of course, you kind of do the best you can to eliminate all the sources that you might, know about. And, but in the nPOP work, we found kind of nicely that there was a limited amount of the variance could be explained from, one sample to the next. So yeah, that was nice to have.
I have a quick second question. Is that when you show the plot of linear scale between protein content and the cell diameter, can you deconvolute if the protein in the nucleus scale similarly to protein in the cytosol?
There's certainly potential approaches for doing those measurements, and there's some students in our lab working kind of exactly on this problem, which hopefully you'll hear more about one day soon. But yeah, theoretically, this involves doing I suppose you could rely on like known information about how much volume the nucleus takes up compared to the cytoplasm. This varies for different cell types. I guess, for example, T cells have much larger nuclei, but in general, this would involve some level of subcellular proteomics, where you're able to measure kind of the protein content in the cytoplasm versus the nucleus to kind of be able to fully get at this question.
Great talk. Thank you, Andrew. To follow up on the previous couple of questions, it seems that there is a substantial interest in figuring out and resolving batch-related effects. Can you please elaborate a little bit more on the actual sample processing, how you place sorted cells on the slide now? Yeah. Can you process them right next to each other, different types of cells, to minimize that type of batch effect? Also, then, how do you transfer or minimize the same batch effects in LCMS? And in addition to that, your negative control samples, do you use the same reagents and buffers, or you also take an aliquot from the cell culture medium where some cells can be those that can be lysed already?
Sure.
How do you account for that? Thank you.
Yeah, lots of good questions. So yeah, I didn't get too much into the specifics of our exact sample prep workflow, but yeah, as there are many, many potential sources of batch effects, one could be that cells are grown in different growth medias, and to control for that, ideally you're able to differentiate the proteomes of cells that are coming kind of from the same sample. So in the course of, for example, some of the data shown from the human testes experiment that Khan is leading, that's kind of just one homogenized, suspension of cells with many different cell types in there. And so your ability to discern different cell types from that reflects, I guess, being able to differentiate proteomes that don't just come from ways that you grow them in culture or different effects along those lines. As far as for our sample preparation method, we are able to dispense, from this cell suspension about 2,000 cells kind of over the course of these glass slides. And so you have, in this case, there could be, let's see, you could have potential batch effects due to, maybe position on the slide. And so essentially the workflow, how it goes is these samples are sequentially picked up and dispensed into this 384 well plate in an automated fashion. And then we're just able to take that 384 well plate and place it in the auto sampler. And so one way to try and deconvolute these batch effects is by running, the first sample picked up and then subsequently with the last sample picked up and seeing whether the variability from run to run is maybe greater than the variability from position to position. And so that's kind of one way in terms of trying to assess because there's always going to be batch effects, but the question is to what extent and how much does this impede you from making the biological inferences that you're interested in. So yeah, there's, there's only so many, I guess, hours in the day, and there's lots of things to explore along these lines. But to the extent that you can kind of use orthogonal approaches to prepare your cells for dispensing, and that you can kind of randomize the order in which you run your samples, this is kind of the way that you're best able to attribute these different sources of variance and try. Once you know them, it's actually, for the most part, of course, there's additional issues, but if you understand the sources of variance well, and your samples are randomized, you should kind of computationally be able to remove most of these sources of variance.
Any other questions from the room? Yep.
Hello, great talk. Also a question about the normalization. I was wondering if you compared the results from with and without normalization, because normally if everything is reproducible without any normalization, the conclusion, at least the biology conclusion should be the same.
Yeah, so actually the person sitting behind you is probably best posed to answer this question. So analysis of we've computed the dimensionality reduction plots with kind of minimal processing of the data. So you can even just do it on the raw peptide level without any sort of these normal batch corrections that we're kind of applying later on downstream, or any imputation approaches that you can, there's kind of there's different sources of missingness in single cell proteomics data. For RNA sequencing, it's all kind of a bit hairy. You have technical dropouts and zeros, but they also could be due to low abundance, and it's a little bit hard to attribute for what. For us, it's a bit more clear. We either have times where the peptide is not detected in the run at all, and this is kind of missing at random values. And then there's also dropouts due to low abundance. And so these can kind of be handled differently, but to the extent you can kind of try to compute these dimensionality reduction plots with and without imputation, and kind of try to see that your results agree. And if you, don't impute and minimally process the data, and, maybe for a PCA, you can compute correlations on a pairwise basis, we're still, for the most part, able to kind of see as one of the main sources of variance the difference between these two cell types, which are quite different. And I guess hopefully you would kind of be able to easily extract that trend.
Hi, great talk. I'm fully with you on capturing the immunophenotype of your cells as you're collecting them. You probably have a lot of experience with this on the cell in one now. Like, how well does the flow cytometry part of it work, and are you guys using it?
That's a good question. We've used it minimally. I haven't done any real work to kind of extensively validate this. We're not using it for quantitative measurements of fluorescence. I think certainly it works well for kind of on-off expression, or things of this nature. But I know they have plans, I think, hopefully to improve or, work on or do some results kind of validating the extent to which you should be recommended to rely on the quantitation from these fluorescent readouts. So yeah, we're not using it in a quantitative sense, like looking at, high or low expression and then mapping that to the single cells. But the image data can be interesting. And in fact, I guess Jason Dirks in our lab has worked a little bit on trying to tie the images themselves to understanding something about the proteome. And again, the quality of these images depends a little bit on how clean your glass capillaries are before you start the sample prep and a couple things. But I think it can be really useful to tie in more of this information, and hopefully in the future we'll kind of work along those lines. Thanks.
Thank you. Great talk. Just to mirror everyone else has said. But I just had a quick question to mirror off a few previous questions in terms of thinking about cell viability, especially within the sample prep process. At least for our lab, we've had long lab days, in terms of just trying to optimize and everything. And have you thought about or can you comment on from the steps from cell dispensing to trypsinizing, how to minimize just cell loss and cell stress and maximize cell viability? Because I can think of a number of projects where that's really important between those two steps.
Yeah. So one of the really nice things about doing proteomics as opposed to RNA sequencing is, for cells to be able to synthesize or degrade proteins, ultimately this requires energy. So to this extent, we are in our experience, we see kind of a less amount of batch effects depending on how we, prepare these samples. We've done tons of experiments where we've taken suspensions of cells and kind of stored them and in liquid nitrogen and then brought them up and immediately prepared them. And to the extent that you're interested in, assessing relative differences between samples and you handle all these samples equally, this can be, a really successful way to kind of go about the handling your samples. But there's also other cases where you're working with more precious samples where this proved to be quite challenging. For example, some project we're working on now using, I didn't annotate this PCA very well, but there's kind of basal cells and ciliated different lung cells. And for this project, we found that we were having trouble with the low cell number of kind of freezing the cell samples down. But a lot of the data was collected just by sorting the cells into a PCR tube in PBS and kind of transporting them on ice. And we've done tons of studies where, the first cell is kind of dispensed to almost an hour after the last cell, and we don't see substantial differences in the proteome over time, over kind of at least in these time scales. But again, I haven't looked super deep into this, but to the extent that we've kind of examined it in our data, proteomics is nice in the sense that we don't see so many substantial effects along these lines. But of course, it's, if you're interested in a specific biological question, I suppose, it's kind of on a per case basis sometimes.
Any other questions from the room? Okay, we have a few questions from the Zoom chat. Nicole asks, reducing the volume on to 20 nanoliters on chip, comparing with the 5 microliters of the 384 well plate, is only the volume of the lysis or enzymolysis reagent reduced? Compared with the two, how much is 20 nanoliters increased?
So I don't exactly, I can really only speak to, I suppose, with any certainty, our kind of sample prep. And So at least for our plate-based method, we dispense the cells into 1 microliter of water. We're then lysing them in this 1 microliter. We add about around a quarter of a microliter of reagents for the digestion, and then another half a microliter for the labeling. So the final volumes end up being somewhere around 2 microliters. For the droplet sample prep that we do on the slide, we kind of digest and lyse these cells in about 20 microliters. And then for the TMT reagents, when we're doing the labeling, this actually works quite well in kind of the absence of all water. So then we dry the samples down, and then we kind of just are adding about 20 microliters of our TMT reagent. So the volumes are kind of similar throughout. I suppose, you'd have to compare the data kind of rigorously to see think about what the kind of different effects are and whether, large or small volumes are going to be suitable for your sample prep needs.
Also from the chat, Stella asks, in the slide about monocyte macrophage differentiation, were those measurements based on DDA or DIA? Have you perhaps compared them?
Yeah, so those measurements were made based on DDA experiments using kind of the scope 2 approach, which uses an isobaric carrier. And I think that's a really great point. To the extent that there's systematic biases in all the measurements that we make, whether it be co-isolation from DDA or interferences from DIA, if you're doing studies that rely heavily on quantitatively accurate measurements, comparing these two measurements can be a really nice additional orthogonal way to compare the accuracy of your data. And to the extent they agree, they can provide you with a higher level of confidence and give you kind of more power to say certain things and have confidence in your data. So yeah, that's a great suggestion. And we've done limited work comparing this so far, but hopefully plan to.